Thanks a lot for the organizers for giving me the opportunity to talk. Genetic variants can have profound consequences for human health by causing misregulation of protein levels. And recent technological advances have enabled us to start looking at the relationship between genetics and gene expression. And I like this slide from Mod ENCODE because it highlights the broad range of gene expression phenotypes that we have started looking at. However, missing from this picture is the fact that every single one of these processes takes place within the nucleus. Yet the journey of the mRNA continues once it's in the cytoplasm, and processes such as translation are thought to contribute to gene expression regulation. Yet little is currently known about how human genetic variants affect translation. Furthermore, we still lack an understanding of how the effects of genetic variation travel through the various levels of gene expression to determine protein levels. So to study this problem, we have turned to one of our favorite models, the lymphoblastoid cell lines. And one of the main reasons why we use these cell lines is the fact that we have genome sequences from these cell lines. So that allows us to look at these relationships. And importantly, the Snyder Lab, we have previously generated proteomics data from about 100 individuals that allowed us to measure the protein levels. And my goal in this study was to really try to link these two by filling the two parts, by looking at both the RNA expression and also looking at translation in conjunction with these data sets. While we are all familiar with RNA-seq, ribosome profiling is a relatively new technology that actually uses a very old idea. And that is that mRNAs differ in the number of ribosomes that translate them, and these differences are associated with differences in translation rate. So the really key step in this protocol is an RNase digestion step. So what we do is we degrade the RNAs that are not protected by ribosomes, and by sequencing the fragments that are protected, we can get a nucleotide resolution sense of where the ribosomes are in a cell. So we started by optimizing this process, and what we see here is a polysome profile which can separate the RNAs by the number of ribosomes sitting on them. And what we have found is the method that's commonly used, that uses a particular RNase, actually does not lead to the expected effect of collapsing the signal into monosomes. And by optimizing this step, we were able to get a very clean signal of the monosomes, and we were able to generate high-quality ribosome profiling data. So using this data set, the first question we start asking is, what are the between individual differences in RNA expression versus ribosome profiling? So as we know, there are many genes that show between individual differences at the RNA level. But what we found was there is only a very few, not a very few, but a small subset of these are actually showing differences at the level of ribosome occupancy. So what this result suggests is much of the variation between individuals at RNA level is actually being lost when we looked at translation. So the next thing that we really cared about is what is the relationship between these personal differences RNA levels and protein levels. So one can ask this question for each gene, and actually for many genes, the relationship is actually quite poor. So if you look at the RNA expression across people and how the corresponding protein levels are, there tends to be lower correlation. But this is clearly not the case for all genes, whereas in some genes there's a better relationship. So if you look at every gene in the genome, the actual median correlation across these genes is a relatively modest 0.2, with only 20% of genes showing statistically significant correlation. So once one looks at the subset of genes that are variable in RNA levels, this is slightly better. But what we found is once you look at the subset that is variable at both the RNA expression and ribosome profiling, then this relationship is much stronger with a median correlation of 0.7, and nearly all of these genes have statistically significant correlation between differences in the RNA and differences in proteins. So the next thing we wanted to ask with this data is, are there genetic variants that specifically modulate translation? So to get at that, what I mean is, are there genetic variants that lead to similar levels of RNA expression, yet lead to differential occupancy by ribosomes? And indeed, we found many examples of these, and we were also able to find certain classes of these variants. So one such class was the genetic variants that modulate upstream open reading frames. So here's one example where the reference allele encodes an upstream open reading frame, and a genetic variant in the population leads to the loss of the URF. And there are different versions of this, and in one particular interesting case, there are two different variants that lead to the same event. And using a standard approach, one can show that these can result in significant differences in ribosome occupancy, but not in RNA expression. And for some of them, there's also differences in RNA expression, but we were able to find many examples where this was just the case for changes in ribosome occupancy. And the another particularly interesting feature that we've looked at is the region around the start codon. So this is the Kozak region, which has been known for a long time to affect translation initiation. But with using our data, we actually were able to look at that for all genes. And in fact, by just looking at the context of the start codon, you can have a significant effect on translation efficiency. And similarly, there are genetic variants in these regions that affect ribosome occupancy. And what we have done after this is to try to validate these with an independent assay. And what we took advantage of is a bicistronic construct where one of the luciferases is translated using an iris sequence, which allows us to normalize for differences. And we cloned the entire 5' UTR in front of the first luciferase with the single nucleotide change, and we were able to recapitulate these differences that we found in our data set. So to summarize, what we have found are that many genes that show between individual differences RNA expression actually do not show this difference in translation. And I think this is important for lots of studies that are looking at bigger cohorts of people in terms of their RNA expression. And it suggests that we need to be careful about what we're concluding by those differences. Which of these differences are really going to affect protein levels? And we were also able to find some genetic variants that are specifically acting at the level of ribosome occupancy and not RNA expression. So to give you an idea of where we can take this project, just want to tell you a very small part of a study that I've done with my colleague Carlos Araya in the lab. And what we have done in this study is, okay, so we now have these variants that can act at the translation level in the human population, and we can ask, are there some disease mutations that similarly are affecting translation in different disease? And one disease that we care about is looking at cancer. So in particular, as you'll remember, we have shown the effect of genetic differences in the Kozak region in regulating translation. So what we've done is we've looked at about 5,000 human cancer exome data sets for such mutations. Are there recurrent mutations in these translation affecting regions? And interestingly, what we found was in a single gene, in a 3-base window in bladder cancer, almost 15% of all patients have a mutation. So it's really fascinating to us because this is in the existing exome sequencing data, yet this has been missed by previous studies because when we look at exome data sets, we generally scan for protein coding mutations and don't really look at these untranslated regions and different places. And more recently, we are trying to build biological models to validate these effects in systems, and we have started to see effects of these mutations on cancer signaling pathways. So with that, I'd like to end and would like to thank Mike for really giving us a great environment to do our work, and I'll be happy to take questions. Thanks.
So with regards to your first point about genes having variation in RNA but not the translation, can you comment on your a statistical power to detect those two types? Do you think it's equal for both or might not be equal?
Exactly. That's a great point. So we have a slight difference in terms of power because RNA-seq is a little bit more better. So the way we have done this is we had replicate experiments for each individual. So that's way we can see what is the error that we get from the measurement versus what is the difference between individuals. So we did, we have slightly lower power, but even when we restrict the set where we have equal power, because for some genes we do, you still see the same effects. And I'll be happy to talk more about it, yeah.
Very nice talk. I'm wondering if you could comment on the potential effect of synonymous mutations. I'm wondering if there's a certain pattern, I'm hypothesizing maybe you could see in this data where mutations that don't affect the amino acid sequence might affect the movement of the ribosome due to codon usage, and whether you see evidence of ribosome pausing actually at synonymous sites.
Yeah, I think that's a great question, and it's something that I would definitely be interested in looking at. However, in this particular data set, we have used so I think the short answer is it is possible, but we have not seen it in our data set, and I think there are some technical reasons. Again, I'll be happy to talk about. So yeah.
Any other questions? Okay, thank you very much.